Assalamualaikum everyone, welcome back to this channel. This is me Ari Wahid and you are on the channel of Medicare. So as you know that we have started the practicals over the pharmacognosy. We have discussed the first 5 practicals. This is the experiment number 6. Within the first 5 practicals we have discussed how to extract alkaloids, how to extract glycosides. So we have studied about the extraction of different alkaloids from different plants. The first alkaloid that we have separated out is from the hyoscyamus seed, second is from the Atropa belladonna, and the last is nicotine from the tobacco. Then we started the glycosides. The first glycoside that we have extracted out is from the nerium leaves, and then we extract out anthraquinones, which is a type of the glycosides, and we extract it out with the senna leaves. Now this is the experiment number 6, within which we are again going to extract or identify, but now not the alkaloids, not the glycosides, we are extracting the starch. So starch comes under the carbohydrate. So in short, we are extracting and identifying a carbohydrate, and the source from which we will do that is our potato, right? So before starting how the extraction do, first focus on the potato, which is our major purpose. So you have to remember the scientific name and the family and the part use of our potato. So the scientific name is Solanum tuberosum and its family is Solanaceae. Chemicals that are present within it is the starch, which is a homopolysaccharide. So it is not a hetero. This starch which is present within the potato is classified under the homopolysaccharide. It contains amylose and amylopectin, which are the both are the types of the starch. So both types of the starch are present within our Solanum tuberosum, right? And its molecular formula is right there. Its linkage is, so within the starch molecule you will find the linkage of alpha 1 4 linkage between the glucose molecule, right? And we also contain alpha 1 6 linkage within the amylopectin. So amylose contain alpha 1 4 linkage, while the amylopectin contain alpha 1 6 as well as alpha 1 4 linkage, right? So this is the basic concept that you know, we have studied it from our childhood. So this is the thing that we are studying right there. So this was the short introduction that what we are studying out and what thing we are extracting out, right? So before starting the experiment, we will see the apparatus that we use that. So of course we need a potato there, we need a knife to cut it out, and we need different lab apparatus that we need here. So I just call the name of them. We need a Bunsen burner, vacuum pump, pestle and mortar for the grinding, weighing balance, and we need a filter paper, and we need watch glass, test tube, and iodine solution for it. So iodine is just like a reagent for it. So always you have heard about the starch, and with the starch you have heard another name which should be always iodine. So you know that there is some relation between the iodine and the starch. Now we will study that what this relation is, right? So before the start of the experiment, first of all you have to weigh your potato, and it should be about 100 gram. Okay, so you will take that potato whose weight should be 100 gram. If it is not there, just cut that potato and just see that it should be 100, right? And you have to peel it off, the outer cover of it, and you have to cut your peeled potato into very fine slices. So you will take your potato and then cut it out into very fine slices, just like a thin slice of it, right? Because we have to grind it out, so you will take your pestle and mortar and you grind this sliced potato. You will put it into your my drawing is so bad right there. Okay, so this is the bowl of our pestle and mortar. You will put your potato slice into it, and then you will grind it out with a sufficient amount of water. And after that you will transfer this potato paste into a beaker which already contain 100 ml of water. So after that, when it become a paste like that, so I am making a paste like this. So this is the paste of that slice of the potato, and then you will add it into a beaker. So here is the beaker which is full with the 100 ml of the water. So this is water and it is about 100 ml. You will add this paste into this beaker, and then you will filtrate this homogenate with the Muslim cloth, or you can use the filter paper here. So we have to now filter it out. So after mixing it into that, so I think I need another color marker to represent. This is the mixture of potato. Okay, and I put this mixture into that with the blue dots. You will already can see that the potato paste is already in there. Then we will filter it out. You will use a filtration flask or something. You will put this into this and you will filter it out right into another beaker, and you will get the residue after the filtration, just like that. Okay, so you can use the filtration flask for it, and if filtration flask is not right there, you can use Muslim cloth, because it is well that easy to observe that, and it make the filtration fast as well. After that, you will titrate this residue again with the water into the pestle and mortar. Again you just transfer it into the pestle and mortar. Again this process repeats. So this process start within a circle. So you will titrate it again with the water within the pestle and mortar, and you will repeat this process 3 times. So these circles travel 3 times. After that, after doing three times, allow the combined filtrate to settle. Okay, now you will allow, after the third time when the filtrate come out, you will allow this filtrate to settle down, and you will see a lot of granules of the starch are being settled to the bottom of your beaker, and you will clearly observe that it should be visible to you, and you will see that. Then you will extract this granule of the starch out. So I am drawing just here. So how you will extract it out, like that, into another beaker. You just take the granules of all the starch out from the potato. Okay, and then you wash this starch so that if any other kind of impurity is still present in it, should be washed out, and your pure starch granule should be there in this beaker. Okay, and after that you will collect a compact mass of your starch. You will filter it out, and then you will put it into the oven that is available into your lab, and you will put it into the oven at the 60 degrees centigrade, and you will allow it to dry. And when it dries, so it become a powder like, so you will get the powder of the starch within you. Okay, after that we have to check it out that whether this is starch or not. So we will perform the iodine test on it, iodine solution test over it. Okay, still now we have performed our first step, which is about the extraction of starch. We have extracted successfully our starch from the potato. Now there is the starting of the step number 2, which is about the identification, whether it is a starch or it is something else. To identify it that it is a starch, we will perform a test which is called iodine solution test. So which is iodine solution solution test. Okay, so for that you will take your 5% starch solution, the starch which you get from the extraction process. This is the starch. You will make the 5% solution of it. So how to make a 5% solution? That is very easy. You will take 5 gram of the starch powder from it, and then you will add it into the 100 ml of the water. So this is the 100 ml of water. You will add 5 gram of the starch into it. This is the 5% solution of our starch, right? And just remember one thing, this water will be hot. Okay, into the hot water you have to add your starch. After that you will add only one drop. With the help of a dropper you will add only one drop of one normal HCl solution to your this 5% solution. And after adding one drop of HCl you will add 2 drops of with another dropper. Okay, with another dropper you will add 2 drops right there, 2 drops of iodine solution into it, and this will appear with the blue color. Okay, so I am using another pointer to show you that the blue color is appeared right there. So this is the blue color after adding the 2 drops of iodine solution. So it means the presence or the appearance of the blue color indicates us that the starch is present there. So it means we have successfully extracted and identified that within the potato the starch is present, and the iodine color solution showing the blue color, which means the starch is present within there. And we can identify the some other material that are also present within our potato. For example, it contains the protein, about 18 to 21% of it, but it consists of 65 to 80% of the starch. So every potato contains 65 to 80% of the starch is present in every potato. Okay, so after that we also have to check the purity of it, like how much purity of we have extract the starch, so how much purity it has, or whether it has not everything is 100% purified, it still has some impurities within it. Sometimes the proteins are also mixed within the starch powder. So to check the percentage yield of it, we have a formula here. Just see the purity of it. Okay, so to detect the percentage purity of our starch solution, percentage yield, or you can call it percentage purity. So you will take the weight of your starch, so the dry powder that you get after the opening the starch, you will weigh it out with the weighing balance you get. Okay, so this is the weighing balance from which you weigh out, and it comes out the weight is 7.8 gram. You will divide it with the weight of the potato. So always you have to take out the weight of the potato is 100 gram. You will multiply it out with the 100, and then you will get the percentage yield, which is 7.8%. Okay, so it means within the 100 gram of the potato you will get 7.8% purity of the starch within it. So this is how you will perform that, and at the result you will show that we have detected it out that amylose and amylopectin is present within the starch, which is responsible for causing the blue color of our iodine solution. So this is how we extract and identify our starch. So here our experiments ends. So within the next video we will start out our 7 experiment, which is just about, I am just telling the name of it. It is about the extraction and the purification of pectin from the citrus peel orange. Okay, so now we will extract out the pectin from our orange fruit, right? So this is all about today's experiment. I hope that you like this video, and if you find this video well, please like, share and subscribe it, and share it with your friends. Take care. Meet you soon in the next lecture with the next experiment. Allah Hafiz.